Behavioral Neuroscience, Chapter 15, Segment 2. Facial muscles can be divided into two categories, the first being superficial facial muscles. These are attached to facial skin and pull on skin and change the shape of the mouth, nose, or eyes. The second is deep facial muscles, which are attached to the bone and enable large movements of the face, such as chewing. Human facial muscles are innervated by two cranial nerves. The first is the facial nerve, which is cranial nerve 7, which receives information from the face and controls the superficial muscles there. The second is the trigeminal nerve, which is cranial nerve 5, which receives information from the face and controls jaw musculature. The facial feedback hypothesis is the hypothesis that our emotional experience is affected by the sensory feedback we received during particular facial expressions, such as smiling. Studies have shown that people who are manipulated into mimicking facial expressions of happiness or sadness experience a change in mood. Impaired facial expressions may affect social interactions. Bell's palsy is a disorder usually caused by viral infection in which the facial nerve on one side stops conducting action potentials, resulting in paralysis on one side of the face. Almost everyone affected recovers within 6 months. Brain self-stimulation is the process in which animals will work to provide electrical stimulation to particular brain sites, presumably because the experience is very rewarding. Effective sites for self-stimulation are concentrated in the medial forebrain bundle, a collection of axons traveling in the midline region of the forebrain. Dopaminergic stimulation of the nucleus accumbens seems to be very pleasurable. Drugs of abuse may activate these same neural circuits. Decorticate dogs were found to respond to routine handling with decorticate rage, sudden intense rage characterized by actions such as snarling and biting in dogs that lack clear direction. James W. Papes proposed a subcortical circuit of emotion because the cerebral cortex normally provides inhibition of emotional responsiveness. Interconnected regions, known as the Papes circuit, include the mammillary bodies of the hypothalamus, the anterior thalamus, the cingulate cortex, the hippocampus, and the fornix. Paul MacLean later proposed that the amygdala and other regions also interacted with this circuit, and he called this whole thing the limbic system. Kluver-Bucy syndrome is a condition brought about by bilateral amygdala damage that is characterized by dramatic emotional changes, including reduction in fear and anxiety. It has a taming effect and induces hypersexuality. Fear conditioning is a type of classical conditioning where a previously neutral stimulus is repeatedly paired with shock or some other unpleasant experience, causing the individual to act fearful in response to the stimulus. The amygdala is a group of nuclei in the medial anterior part of the temporal lobe. Lesioning just the central nucleus of the amygdala has the same effect as a lesion of the entire amygdala. A direct projection from the thalamus to the amygdala, called the low road, bypasses conscious processes and allows for immediate reactions to fearful stimuli. An alternate high road pathway routes information through the sensory cortex and is integrated with a higher level cognitive process. Contributions from the prefrontal cortex and anterior cingulate gyrus offer an additional level of fear conditioning called observational fear learning, in which fear is learned through social transmission. Information about the stimulus from several brain regions reaches the lateral portion of the amygdala first, and evidence suggests that neurons here encode the association between specific stimuli and aversive events. The central nucleus transmits this information to various brainstem centers to evoke 3 different aspects of emotional responses. Number 1, pathways to the periaqueductal gray evoke emotional behaviors. Number 2, pathways to the lateral hypothalamus evoke autonomic responses. 3. Pathways to the bed nucleus of the stria terminalis evoke hormonal responses. Different types of fearful stimuli may activate slightly different pathways to and from the amygdala. Activity in the prefrontal cortex is necessary for an individual to benefit from fear extinction training. Evidence suggests that stimulation of cannabinoid receptors normally extinguishes learned fears. This suggests that medications that target these receptors could be created for phobias. Blood flow to the amygdala increases when humans are shown images associated with fear. Evidence suggests the brain relies on other symptoms to detect internal threats, such as lack of oxygen. Patient SM, whose cells in her amygdala have been destroyed by calcium deposits, can still become afraid by sensing internal threats such as air with a high concentration of carbon dioxide.